Again, the organ is a lung. What we observe here as a pulmonary structure, that here is a vessel which is a little bit smashed. Also, what we observe here is pulmonary and eventually alveolar parenchyma, which is suffering severe atelectasis because it's pushed by something which is causing pressure, a volume process. Some small portions of anthracotic pigment again, nothing more specific. And there are some hemosiderin latents, which you can observe here due to the stasis of blood. This brownish pigment here is hemosiderin, which is sometimes found in macrophages, and you can observe them well. Further on the slide, we start with this zone specific, which is actually a formation of a tumor. This tumor is not well circumscribed, and you can see that there is some hemorrhagical changes at the periphery of the tumor. We see some hemorrhages, edematous changes, inflammatory infiltrate at the form of stromal reaction, which is made of, what are these cells, leukocytes or lymphocytes? Great. Yes, stromal reaction is majorly due to accumulation of inflammatory cells at the periphery of the tumor, which are trying to demarcate the tumor growth and the tumor process. So what we reach is a further region, which is made of cells which are completely identical to each other. They're looking like clusters of nuts and nests of cells which are not well demarcated by fibrous tissue strands. You see that the stromal reaction here is very actually spared. It's not much in between the nests of cells. And if we enlarge these cells, we can observe that there is a specific manner of arrangement of chromatin inside in the nuclei. It's at the form of salt and pepper clusters, which is due to the chromatin arrangement here. Let's check out for, oh, it's gone again. Sorry, guys. Что-то ко мне исчезло, гадостно. Sorry, guys, today the connection is bad. I'm really sorry for which. I don't know why all these stuff are disappearing. Maybe they're too big for explanation here. Anyway, so what we observe here is majorly same arrangement of cells which are pretty much appearing identical to each other. No evident nucleoli inside, just clusters of chromatin in hyperchromatic nuclei, which is actually having the arrangement and the manner of arrangement of salt and pepper. You can see how it's looking granulated, as if there are lighter fields inside the nuclei and darker fields, as if salt and pepper mixed together. So what type of tumor is that? Not much stromal reaction, nests and sheets of cells, small cell carcinoma. So where did that start from? Yes, it's definitely a carcinoma, but what type? Yeah. And what were the features of tissue and cellular atypia here? Cellular definitely needed to be mentioned here about this tumor and about all types of tumors. So remind me, what were the features of cellular atypia now? Pleomorphic cells? Well, not exactly here, but yes, this is a feature of cellular atypia. Hyperchromatic, definitely. You can see how the field here is much darker than the others. Well, naked cytoplasm, ratio of the nucleus and the cytoplasm, yes, it's definitely different. What does that mean? That the nucleus is much bigger than usually we expect to see it in normal cells. Increased mitotic figures, yes, we can observe them on a high magnification very well. Let's check out. Oh, how is it gone again? I can see it well. Do you see it now? I see it well on your screen. Okay, thank you. So, what else? Where does this tumor start from? Yes, completely correct. What does neuroendocrine mean? Which are these cells? We discussed them in practicals a couple of times. We discussed them also when we had practical about pulmonary system, also in gastrointestinal system, and in other spaces and in other practicals where we had the component of neuroendocrine tumors. Which is this system, a specific one, with scattered cells in between organs, in different organs. This is the amine precursor uptake decarboxylation system, which is bringing some scattered cells with neuroendocrine function and secretion of specific, well, hormones, majorly somatostatin, which is connected to different regulative functions in the organism. And these cells could give rise to tumors, which finally could be looking like this. There is a very detailed classification of neuroendocrine tumors, which I don't believe that you need for your exam. For this type of tumor, you need to mention just that it's actually, it's gone again. Sorry, I will share it again. I think you can see it now. Yeah. For this type of tumor, you need to mention that it's a small cell type of carcinoma in the lungs, a neuroendocrine type of tumor. If it's a tumor or a carcinoma, it depends on the mitotic count and the proliferative index that we can observe on the inside of the cells and in the nuclei. And majorly, we should mention that these cells are identical to each other, forming huge nests with not much of stroma in between the nests of the tumor cells, and with very typical characteristic of the nuclei with a dispersed chromatin in the salt and pepper manner. Do you have any questions about the slide? Something that you cannot understand? Something not clear about the slide? Here is the tumor again. Here is again. Here it is again. Our suggestion is malignant. Wait. Let's check out for the box. Definitely a malignant one. This one is showing many mitotic figures in here. It's not well demarcated. It's huge in size. Yes, the whole dark area here, which is pushing the surrounding structures of the lungs apart, and it's causing atelectatic changes inside in the lungs. This is all connected to the malignant tumor that is growing here. Yes, the whole dark area is a tumor. So, any other questions? No. Now we'll continue with the next one.